Hi everyone, my name is Junyu Chen from the Biomedical Electronics group in University of Southampton. Now I will present my previous work on the electromechanical shear flow cytometry for single cells electrical property and the deformability analysis. So here shows the summary, the background, the end of our work. The figure on the left compare the current single cell analysis techniques. These techniques provide a way to phenotype cells based on physical properties that enable different cells population to be characterized and distinguished. The traditional method like the AFN and the optical stretcher and others provide relative high precisions, but they are really low throughput and need high technical capacity. So the microfluidic method significantly improve the throughput of the technique. The first one is the context-based methods have limited speeds, while the extensional flow-based, the FDC, achieves really high throughput, but introduce the additional system complexity and reduce the accuracy of the measurements. So we made the balance between the two methods and try to decide to develop a shear flow-based methods to development a new single cell analysis methods to measure the electrical and the mechanical property simultaneously. So it's simply to measure the impedance of the cells at multiple frequencies using specific electrode arrays. And then extract the key parameters of single cells like cell size, stiffness, and membrane capacitance. And there are some advantages of these measures. It's really fast and high throughput with only small sample values, and they can simultaneously measure multiple parameters, and have a really high accuracy and a low cost because we do not need the high-speed cameras, and also less complexity because we do not need the chef flow to concentrate the cells, and also do not need the cost channels shape of the cytometries, and also can enable the real-time monitoring. Here I explain the working principle of the systems. Cell will first suspend in the viscoelectric fluid buffer and then flow through the microfluidic channels and experience the shear stress and deforming in the narrow channels. And the viscoelectric fluid also partly focusing the cells into the central region to reduce the positional dependence of the cytometries. And at the same time, the two sets of impedance Z1 and Z2 were measured and recording by the microelectrodes arrays at different frequencies. And the first set of impedance Z1 is measuring by the electrodes produced the orthogonal electrode fields along the flow direction, and the other set two is measured by the same direction of the fluids. So both impedance can provide the change of the cross section of the cells due to the shear stress at different and two different directions. And the high frequency impedance and provides the electrical property of cells like membrane capacitance. And the simulations show the electrode deformability defined as the ratio between the Z1 and Z2 is almost independent to the particle size and is linearly proportional to the optical deformation index, the major SS to the minor SS, the ratio of the major SS to the minor SS. So for the real system verification, we use three different ways to treat the cells. One is the osmotic stress to order the cell's size and the stiffness at the same time. And the second way is to glutamathize the cells to make the cells stiffer and make the cytoskeleton destruction by cytokeratin D to soften the cells. Here show uh, the system the company system overviews. Include the microfluidic impedance chips MIC and a series of other components. Uh, the MIC chips contain a narrow measurement region which integrates electrodes and also include the optical window for the cells images captured. And the chips will be inserted into two PCB boards and a holder. And during the measurement, a mixed AC voltage will be generated by the lock-in amplifier and then applied into the microelectrodes. And the orange node output is then amplified and demodulated and transit to the computer for further data processing. So finally, we can get the required parameter like electrical deformability, electrode diameter, and the opacity. And here we can see the raw scatter plot of the cell phenotyping and optical correlation results using the HLCT cells. So the scatter plot shows the relationship between the electrode diameter, opacity, and the electrical deformability. So be noticed that all the electrical parameters are normalized by the rigid bits, which is the reference for the different group comparisons. And you can see the figure, the small values of opacity indicate the higher membrane capacitance, while the large value of the electrical deformability correspond to the lower cell stiffness. So the undeformed objects like the rigid bits have an electrical deformability around 1. And on the right, the optical images confirm the cell deformation under different treatments, like the glutamic fit and the cytokeratin treatise. And the graph showed a strong correlation between the electrical deformity and the optical deformability, validating the system is working well with higher accuracy and the sensitivity. So here show the experiment result of different treatments. For the glutathione cells, the electrical deformability decreased, indicating the increased stiffness. So while the cell size saw no significant change, and the opacity increased, suggesting the reduced membrane capacitance. Reduced and the increased membrane permeability. And for the cytokeratin D treatment, electrical deformability increase, indicating cell softening, while the cell size and the opacity show no significant change. And both these half maximum concentration are in good agreement with the previously reported values. And the last one is the automatic shape with the cells treated with different osmotic shape. As osmotic increase, cell volume is decreased as shown in the figures, and which reflects by a decrease of electrical diameters. And the electrical deformability also decreased with the decrease of cell size, while opacity showed a decrease, also saw a decrease. So indicating the cell membrane capacitance increase. So all these results demonstrate that the system can sensitivity detect the change in both the mechanical and the electrical properties under different conditions. So here, I have demonstrates a microscope Shear flow cytometry platform to enable to simultaneously measure the single cells, analyze. Yes, thanks for your listening.
So, as Bastian said, I would like to present today our recent work on the design of materials, especially for 3D and 4D printing with high precision. So we are at the University of Heidelberg in the southwest of Germany, in a very beautiful city, also one of the oldest university in the country. And we are working in a new institute, which is called the Institute for Molecular Systems Engineering and Advanced Materials. So it's a long name, but the peculiarity is that we work together, chemists, physicists, material science, biologists, and engineering, with the idea to have a very interdisciplinary environment. And yeah, our group is focused on the design of new functional materials that we can save with light. So here is an overview of our activities. So we go through the synthesis of new materials with a special focus on the photochemistry. You will see later, we print with light. So all the photochemical processes play a big role. So starting from the synthesis, we go through the characterization and of course also the processing of these materials with light. Due to my background, we work also a bit on self-assembly. I will show a couple of examples. And lately, we have also introduced sustainability aspects in the design of, and processing of our materials. So one of our main focus is to apply these materials of 3D printing or additive manufacturing. I think I don't need to give you a big motivation about that. So 3D printing at the beginning, of course, it was more about prototyping, but now it really made to the applications. Here an overview in medicine. For example, if you have been recently at the dentist, so it's not anymore about molds, you can personalize all of this at a big advantage of additive manufacturing. Also, we have collaboration with the clinic here in Heidelberg. So it's also very practical to print in advance the models before an operation so that the surgeon can practice. Sport, an example is Adidas shoes. Food is more, I would say, as a hobby, but you see, so there is a wide variety of applications and also a wide variety of techniques. And as I mentioned at the beginning, we focus on light-based additive manufacturing. And let's start with the basic. I will be too brief, but just in case someone is not familiar with that. So let's start with the basic of the photochemistry behind this process, the difference between one photon and two photon processes. So you know for sure the one photon absorption process, a molecule absorbs one photon and goes excited from the ground state to the excited state. This process can be also done by the absorption of two simultaneous photons. So you see here that, of course, there are different implications. The first and most obvious one is that the energy is different. So these photons, they will have less energy, meaning that we think also double of the wavelength for the two photon absorption. But also another important implication is the probability of this event. While the probability of the absorption of one photon is relatively large, in the two photon process, we need the simultaneous absorption of two photons, same time, same space. So here the probability is lower, and this event will only happen where our laser has a high dose, so in the focal point. So one very visual way to see this is with emission, with a fluorescent dye. So here we have a cuvette of a fluorescent dye. If we excite by a one photon process, you see the emission on the optical pathway in a larger area. While if you do by a two photon process, you see that you need to zoom in. So it's only happening in the very small volume. This is of course used in microscopy for high resolution, but now if we, instead of a dye, we exchange this dye by a photoinitiator, now we can initiate by a one photon or by a two photon polymerization, and that's actually what happened behind the 3D printing of polymers. So in the process of stereolithography or digital light processing, so macroscale printing is a one photon polymerization process, and for two photon polymerization process, we have the advantage that we can go to the micro and nanoscale. So we have this polymerization very precisely focused, and we can achieve high resolution. So how does it work? I have here an animation. So we have a liquid ink, and I will talk about the details about this ink behind. We have a femtosecond laser, usually in the infrared, is focused inside, and you can imagine that this is a pen. So now we polymerize, so the material solidified, and we can, in principle, print any arbitrary geometry with submicron resolution. The applications I will also, depending on the material, I will emphasize a bit, but in general, it can go from photonics, microfluidics, micro robotics, and recently we have also explored a bit applications in the life science region. Okay, so a bit of overview about the history. So indeed, the first printed structures, they were already in around 2000. So you see here, these small structures. At the beginning, it was more a technique used by physicists with some commercial materials. I think the real breakthrough was with the commercialization of this technology. And recently, this really made a huge advance technology, so it's not anymore so slow. You can parallelize, you can really print fast with very high resolutions, very smooth, and with a lot of advantages. Okay, so that's all about the technology. I'm a chemist by training, so what we have been doing recently in my group is to use this technology and expand the library of materials. And as you have seen, there is also a wide variety of applications, so we need to also match the materials. So if we focus on the fundamental, what do we need? So we need monomer. If possible, for increasing printability, this monomer needs to be multifunctional, so to achieve crosslink. And we have said that we have this small volume, so it's beneficial to have a high density of cross-linkable groups to have good printability. And very important also in the formulation is a photoinitiator. So we need to initiate this process. Very often we use radical polymerization and of course a radical photoinitiator. And as I said, today this talk will be not about commercial materials, but new functional materials that we have developed here in-house. Okay, so I have divided the talk in between two parts. First, I will focus in the 4D printing, and then I will go at the end a bit on the sustainability aspects. So let's start with 4D. So what do I mean with the fourth dimension? So the fourth dimension is time. So that means that we print microstructures that are not static but dynamic, that we can change one property. This property can be shape or other property with time. How we do this? So as I said, we focus on the light-based technology, and the trick is to work with responsive materials, in this case, responsive polymers. And we have tackled three different classes of materials. The first class is hydrogels. So hydrogels are very hydrophilic networks, so they contain a lot of water. And by playing with the chemistry of the monomer, we can make them, for example, thermoresponsive or pH responsive. That means that when we stimulate them with this external stimuli, we change the hydrophilicity, so they will become hydrophobic, they will expel water. So that means that change the optical properties and the volume. Another class of materials are liquid crystal elastomers. Is again a network, and in this case we have liquid crystal. Very important is the alignment, we will see later. And again, this relies on a transition from the liquid crystal to the isotropic phase, changing also the volume and inducing an actuation. And the third class that I will talk also are shape memory materials, so materials that we can program in one shape and then they will recover. So let me start with the hydrogels. Hydrogels, we have said, very hydrophilic materials. They are mainly in water and very important for more bio applications. So we started a few years ago introducing this material in the microscale. And the first example was the well-known NIPAM, poly-NIPAM. It's a thermal-responsive polymer. So we print the NIPAM with a bit of crosslinker. We create a hydrophilic network. And when we go above the low critical solution temperature, it becomes hydrophobic, it shrinks. And with this case, we can do the actuation. Now we have the advantage that we can play with the capabilities of two-photon laser printing, meaning that we can on demand save the material. So what we did at that point with Mark Hipler, now a co-worker of Nanoscribe, was to use these capabilities and print a structure, so starting from the same material, from the same mixture of monomer and crosslinker, we play with the laser power, which some people call it gray tone lithography, and we print a pillar, here in blue, very highly crosslink, which is so crosslink that is not responsive, and then a bilayer, a beam, that it has two different powers. So that means that when we use lower laser power, we will have, of course, lower crosslink. When we have higher laser power, we have higher crosslink, and that has also an influence in the response. So the lower crosslink will shrink more when we increase the temperature, and we can direct this movement of the beams. This is what you can see here in the video. Hopefully, yeah, exactly. So when we hit our water bath above 40 degrees, which is the low critical solution temperature, you see me. Again, we can exploit even further. We don't need to do only a simple beam. We can do also some segments, and then we can play also with the morphing of the beam. At a later state, we have also incorporated, in collaboration with a group here in Heidelberg, the Hywel group, this type of materials inside microfluidic system. So just placing pillars that they will later shrink. One can use it for size sorting spheres or even cells. We have also played with other type of chemistry, especially with host gas systems in collaboration with the group of Martin Basmaya at the KIT. And another capability that I have not mentioned so far of this technology is that you can go to multi-material 3D printing, meaning that you can have structures made of different kind of materials, like the example here, where you have some more rigid, here in gray, rigid supports. Here in orange, you have a material that is cell adhesive, very important if you want to study the behavior of cells, and here's the responsive system. So that means that when you sit after printing the cells, they will sit on top, and with the responsive system, you can stimulate. With another collaboration with biologists, we went a bit further, and we just not print and put the cells inside, but in this case, we inject biocompatible ink inside an in vivo fish embryo. So here it may be a bit small for you, but we inject it in the eye, and we could print inside. So here you have a small star. This is, of course, at the beginning very proof of concept, but idea behind is in transparent systems such as this small fish, you can think of even on in vivo or in organism implants. Okay, so now we have been focusing on hydrogels all in the water medium. So what happens if you want actuators at ambient, so in air? Hydrogels are rather a soft material, and of course the mechanical stability is a bit constrained. And one advantage of the liquid crystal elastomers is that they are very nice for actuation, and also the mechanical stability is better in air. So here, you remember at the beginning, we saw that for the actuation, we need alignment. And here we started formulating or using a standard mesogen. So that's liquid crystalline molecules that they have the printable groups. And here we have a solvent, I will talk more about later, which is also a liquid crystal. So if you have a cell that all the molecules are aligned in one direction, that has been done before, is very nice, but your actuation is limited to one direction, obviously. Here again, we wanted to make it more complex, and here Alexander, PhD student of Professor Begner at the KIT, construct this sample holder with electrodes that allowed us to align while printing. Sorry, it's not working, the animation. Okay, sorry for that, it's not working, the animation, but how it works is that, yeah, now it's working. So how it works is that you apply the electric field, then you can align, and then during the polymerization you fix it, and in principle you could change as many times as you wish the orientation of the liquid crystalline and then fix it during the polymerization. And we did with different geometries. That's here one example of one structure that it has until 9 different orientations. Then you see when you hit up, we introduce this transition, and then we have this vent also working with this bilayer. So that was a first example working with this. You see that the temperature you need is rather high, so not compatible with many applications. So then we decide, okay, why we are not changing the stimuli? So temperature is rather global. Let's try with light. Okay, so this sounds very simple. One can have an absorber right inside the ink, and then one can later induce this photothermal effect. So it sounds easy, but from the point of view of doing it, it's not so simple, and Li fight a lot for that, because of course we are printing with light, so if we have an absorber that's also interfering with the photopolymerization process. And some of the absorbers may not be compatible with the liquid crystalline phase. So that was a bit of a struggle. But you remember that I told you that we have this solvent that is also a liquid crystal during printing. So then Li Yun came up with the idea, okay, why don't we stick to this process that is working? So we have a very well-established protocol, you remember before, where we can print the solvent, and then in the development process, so when we wash the unreactive material, we exchange the solvent by a dye. And like that, we are not constrained, because this is happening after printing. In the washing step, we are not constrained with width wavelength, and she could incorporate the standard isobenzene, absorbing in the UV or blue, but also other DASA and other dyes more into the infrared. And like that, we have all the freedom. We can have different colorful actuators, and here we have the advantage that we can directly actuate it precisely in the spatial and temporal control. And here also with the group of Martin, we made also more complex metamaterials. So you can see there that you can actuate. Okay, so now let's move to the third class. I said safe memory polymers. This class has been really explored a lot at the macro scale, and here in 2022 we tried to also transfer this to the microscale, sorry. And so here, the composition, without going too much into the details, we rely on this main chain builder. Is an acrylate that it creates polymer chains that are in the gaseous state at room temperature. And when we heat above this glass transition temperature, we go to a more rubber-like state, so we can deform our material. Then we can cool down, then it fixes the program shape, and if we heat up, it recovers. So we started because that material was new for printing at the macro scale, and here you can see how does it work. So we print this infinity ring. You see that is not fitting in the little bottle, and we have here a water bath that is preheated at 80 degrees to make sure that we are above the glass transition temperature of our material. When it's at this temperature, it becomes rubber-like, so you can just with the tweezers deform it. Now with this water bath at room temperature, you can fix it. So now it's in the glassy state, and now the program shape is compressed. When it gets inside the bottle, you see that it immediately open and recover. So once we proved that the system has this same memory effect, then we went to the microscale, and then you see we printed the same material several orders of magnitude smaller. So these little things here, we program it by compression. Also these micro boxes where we can, for example, put something inside and then open and close. And just to show you that this is really reproducible, this is a top view of these boxes. So you see that you can compress and then it will recover with temperature. This is a robust process. As soon as you don't do anything wrong with the programming, you can repeat it several times. But as mentioned before, when we work with temperature as a stimulus, then it's a global stimulus, so all will be recovered. So then we came back to this absorber. So can we use light as a stimuli? So here is again going back to the macro scale for demonstration. We have our formulation and we have an isobenzene as an absorber. So we have print this flower, we program completely close, and now just using a laser, we can have a spatially resolved and controlled recovery. So it's not anymore just opening globally, but we can decide which specific point we want to activate and recover. And the same we can with just laser pointer, we can also open, for example, a box that it was compressed. This can, of course, be translated to the microscale. We have not done it, but it was more a proof of principle. Okay, so these were our first steps in this 4D microprinting. We explore this variety of materials, and then the question, okay, what's next? So just to be honest, that's very nice to work with this conventional stimuli responsive, but if you remember at the beginning, I told you they are completely different materials, but they have something in common. They rely on this transition, so you can see them kind of an on-off system, which is enough for actuation, but sometimes you don't want to have on-off, but rather control gradually the properties. So that for that, they are not good enough. And then we took inspiration from nature, from living systems. So the living systems, they can grow to trees. We as a human, if you have a baby at home, then you realize also how fast as it goes. You can, so system, they grow, and they also change the mechanical properties of the tissues. Then we started with a very simple system. Can we do this chemically? So we know that, for example, this polyethylene glycol diacrylate can be very a nice cross-linker in the presence of photoinitiator. And here we introduce one novelty. We introduce this tempo monomer. So you may know tempo is a radical scavenger, so sometimes it's used as a stabilizer. Here we push a bit the conditions and we use a special tempo that it has also a photo cross-linkable group. So meaning that we can incorporate the tempo covalently to our network and we trap it as an alkoxamide. And here now we can play with the alkoxamide chemistry. We can use it as an anchor point, as an initiator, and using nitroxide mediated polymerization, we can make this expansion or growing of our network. Or we can also use dynamic covalent chemistry. So with an excess of tempo, we can do this exchange reaction. We can play with the cross-linking density. In other words, now we have more freedom. We can tune not only the one mechanical property, but we can tune size and mechanical properties in parallel. So here's the chemistry behind. I told you we trap the tempo, then we have the alkoxamide, then with an excess we do the exchange, and we can use this as an initiator point and grow or polymerize, for example, styrene. So here we chosen styrene for two reasons. First one is that we start with polyethylene glycol that is rather soft, and polystyrene is stiffer, and also polystyrene, is a commodity, it's a very important polymer, but due to kinetics, it's something that you cannot print so easily with light. So that was another advantage. So we prepared the formulation, and I show you. This was one of examples of a printed structure that Christoph did, this micrometric gecko. Then we put it in a present of more tempo. So you see that apparently nothing changed because we still have a lot of covalent network, so a covalent link. But we have a small fraction of these opening bonds, meaning that we decrease a bit the cross-linking density. We see a small effect in the Young modulus. Now we have other systems that will have more effect, but what it was more impressive is what happened when we do the polymerization with styrene. So you see this is our initial gecko. After 2 hours, after 4 hours, it's growing because of this expansion, but not only growing, we can increase the Young modulus from megapascal to gigapascal due to the presence of this polystyrene. This is just not only happening with gecko, we have other geometries, flowers, octopus here, that's a GIF what happened. Of course, after some point, then is getting too uncontrolled and then you lose the fidelity of the shape. But after a few hours, that's still keeping quite nicely the 3D shape. Okay, let's go a bit down with the scale. So far, I showed you that we have under control by printing the microscale with a 2-photon laser printing. But what happened to the nano and molecular level? So for the chemists, you may have realized that we like to use a lot free radical polymerization of acrylate, which is very wonderful for printing acrylates. I love them. They are fast kinetics, but they have one disadvantage, is that you have little control of the network topology. And again, taking inspiration from nature, we know that the functionality of the natural system is so great because you have this hierarchical order. You have under control from the molecular level, nano level to the macroscopic level, all is completely hierarchical, and that gives also the functionality. So again, continue with inspiration by nature, we started exploring other materials, and one example is collagen. So you know, collagen is a protein very abundant in living systems, that it can form these collagen fibers. If we go a bit down with the structure, we go to the microfibrils. The microfibrils, actually, they are made of this triple helix of the collagen. And that's all because we have under control the molecular sequence. So this sequence of amino acid made this hierarchical order. So this triple helix, we can disassemble using temperature. So in principle, one can think, okay, that's kind of undesired, right, to unfold proteins. But here, Philip came up with a nice idea to make it useful from the 4D printing point of view. So you see temperature to play with the shape and properties of collagen. So what he did was to make a formulation, a printable formulation of collagen. This is collagen functionalized with methacrylamides groups, so that allows photo cross-linking. One can print a structure, and here it was very important to control that we are not disturbing the triple helix of the collagen. So that was the trick, to make the ink in a way that we are not disturbing. So we have this triple helix we fixed during the polymerization, and now with temperature we can unfold, and then we see great effect in the size due to this shape. This has also been proved by simulations. So here what you see more in black is a frame with a non-responsive material as reference, and a buckyball, a hollow buckyball printed with the collagen. Due to the large presence of water, the contrast is a bit lower. When we increase the temperature, we unfold, then we have this compression, and it's completely reversible. So here we are not destroying the protein, we are only folding and unfolding. And you see in the video, when we go above these 40 degrees, then it contract, and then it completely expand again. We have other geometries, so that works really nicely and reversible in water. So taking inspiration again of natural systems, we have also played with more synthetic systems. Block copolymers, they can also self-assemble at the nanoscale, and by playing with the length of these polymers, you can also fine-tune the type of nano assembly. So we have some of them that they can align in lamella or cylinders, and we have proved that we can maintain this nanostructure inside printed structure. So these were ducts that they were cut and analyzed inside, so we can keep large order inside. And going through the sequence inspiration, DNA, all the proteins, they have the sequence defined. So we have made a smaller oligomers of 8 units where we have changed the position of the photocross-linkable group, and then we see that actually this has an impact on the printability. So this is the alternating one. This is the one that we have them more separated, and then you see that the alternating, this is a typical dose test. They have what we call this printability range in green is larger than for the other ones. Okay, so in the first part, I have focused more on this functionality, on the 4D printing, responsiveness, and molecular design. And I would like to use the last minutes of my talk moving towards sustainability. So the 3D printing is itself by itself a more sustainable printing than other manufacturing methods, such molding or subtracting, because we use only the material we need, and then in principle one can use the rest for further prints. But what we have focused is on the sustainability from the chemistry point of view, from the material side. So you have seen again acrylates and some synthesis that we have done, and we rely always on petroleum-based materials, on petroleum-based feedstock. So what we start doing in the master thesis of Clara was, can we replace this petroleum base that we always order monomers or starting material from sigma alderates? Can we change it and use natural systems? We started just going to the supermarket, and we bought different class of vegetable oil, soybean, sunflower, canola, and sesame, and olive oil. So what is inside the oil is mainly a triglyceride molecule with some double bonds. Unfortunately, we cannot print them directly, so we need to do a formulation where these double bonds are activated with acrylates, that's what you see here, and we need a bit of photoinitiator, but it works really nice. We can print these beautiful structures. These were printed by DLP at the macro scale. We have compared with commercial materials. These are this resolution tests. So you see them, they have a bit of color because they are coming from the natural product, but you can print quite nicely. Also 3D geometries. That was the nice back cover that Clara did. But of course, that's not sustainable enough. We don't want to compete with a feedstock that they are used for the food chain. So we went a bit further investigating alternatives to this vegetable oils. So where can we extract in a more sustainable way without competing with food triglycerides? And here it came the idea to work with algae as an alternative. So here we don't compete so much. I mean, of course, algae, one can eat algae, but it's not, I think, the main diet of most of us. Also, they have the advantage that they have higher growth rate. They can wait even grow even with wastewater. And also main advantage from the chemical point of view is that these double bonds that are the active grade, so they have high saturation degree than the vegetable oils. So we contact here the Bank of Algae in Spain as a collaborator. We discuss this. We really screen different types of microalgae. They cultivate them, and we selected two as a promising one because they were growing very fast, and also they were accumulating a lot of lipids, which is what it was interesting for us. We got the biomass, we extract the lipids, and we functionalize them again with acrylates using the protocol solvent-free. And here, when we were extracting, as you see here in the image, we were getting a green oil. The green was coming obviously from the chlorophyll, which at the beginning we were trying to remove, but like that you lose yield and is of course less sustainable. But here, there's something nice happened. is that we were just printing with the green oil. Without photoinitiator, we were able to print relatively decent microstructures. And maybe the people working more with bio applications know that very often excess of photoinitiator or things like that can be toxic for cells. So here we have the advantage that we don't need it. And again, in collaboration with Professor Basmaya, we could prove that this has also low toxicity. So we can grow cells around as a scaffold very nicely. We have an alternative to vegetable oils, and of course, we will continue exploring other alternatives. So again, so far we have focused on having bio-based materials, but what happened with the end of life? If we want to have additive manufacturing really going to real applications, so we also need to take care of the material after the end of life. And one of the problems of working with, again, this free radical polymerization of acrylates is that they print wonderful, but you end up with a thermoset, something that you cannot recycle. So we have also recently published alternatives, but I would like to show you here one example of another strategy, which is starting with bio-based. Okay, that's already a good step, but also having a network that we can chemically cleave. And here we can, by cleaving using aminolysis, we can get one product that is useful for other syntheses, or in our case, we can use it as a precursor of our monomers. So we incorporate kind of a closed loop. He is not completely closed loop, and there are people really working on that, and that's one of the challenges, to have completely closed loop. So ideally, to have bio-based material that you print and then you recover directly with as less synthesis and chemical processing as possible. Good thing here is that we can print very nicely. You see that there are not much difference between first and second generation, which is one of the challenges, and we can also keep the high performance of the material. And with that, I would like to conclude so that we have some time for questions. So I have shown you how we are designing materials that they have this 4D response. We have been a step further, going towards this, what we call lifelike behavior. It's a bit more complex than on-off systems. More fundamental investigation from the molecular point of view is to not just focus on the network at the microscale, but achieving control at the nano and the molecular level, and some sustainability aspects. So what coming next? Especially for bio applications, we are targeting biocompatibility, multi-responsiveness, and adaptivity. We are in several initiatives working on microimplants and robotics. We continue with the more comprehensive understanding and rational design, because that helps us better to later design more complex systems. And as you have seen at the end, we are also very interested in having more sustainable systems, and we are inside a European network called GREENS. So just to close up, you have seen that very often functionality and sustainability are targeted separately. And what we want to do in the future is to have sustainability, but also while keeping the functionality. So we want to marry the two topics. And with that, I would like to thank again the organizers for having me here today, the group for the great work. Without them, of course, won't be possible to show you so many things today, the funding agencies, all of you for your kind attention.